All right, we're back. Finally. Let's see if I can do this. So I have to do it like this and just scroll through them before. Okay. So to start with, I touched on it earlier. Lining tissue, it's like a sheet of cells. So if you can think of whatever a good analogy would be, a quilt, where all the little patches are sewn together. So they're all not only they're attached not only, let's say, to the bed, assuming it's a quilt of some type, but they're also attached to one another. Okay, that's the idea of what a sheet of tissues of cells are. So they can be relatively thick, or most times they're fairly thin, and basically as a lining cell, it's just that. Most of the lining cells, their role is relatively a smooth, moist coating. You'll see me do this 1,000 times. So it's like something can roll over it very easily. That's how lining cells are. Most of the time, they're in the last lecture, in the last lab, we talked about those body cavities, the pleura, the pericardia, the peritoneum. All of those structures have a single cell moist lining. And that's what these things are supposed to move easily over them, not adhere to them, relatively speaking, etc. So when we looked at that, and definition wise, a tissue is a group of similar cells that perform this function. Fancy name is histology, is where we study them. Cytology is where we study cells. So covering cells are epithelia. That's today's topic. Probably in the amount of time it'll take me to cover it. So the epithelial or lining cells are just, and I am a nut for them. Why is that? Epithelial cells are as actively mitotic. We replace them constantly. Your skin, every 30 days, recycles itself on average. Okay, the cells in your gut, stomach, small and large intestine, every 3 to 6 days, you get a whole new set. Not all at once, continuously, billions of cells that are done like that. So understanding those cells, where we find them and what they do is really important. In every tissue, by and large, different organ systems have certain cells that are hallmark, that we only see in that system with certain adaptations. Because they reproduce so rapidly and they reproduce all the time, it's a good news, bad news kind of situation. Here's the good news: if they're damaged, think of what you put into your mouth or in your gut. They're abrasive, sometimes they're chemicals, sometimes they're irritating, sometimes they might be poisons that have to be dealt with, things that are potentially toxic. So it's going to damage those cells. We make more. Fill in the gaps. Useful. Fall down and scrape your knee. Those are epithelial cells that, soon after whatever the healing period is, and the little scabs go away and all that stuff. Down, you don't even see that. I don't remember where it was. It was down here somewhere. Good news. We don't repair ourselves so much as we replace them. The difference is, okay, that because of that, the fact that they're so vitally reproductive, things can go wrong in the replicative cycle of mitosis. Errors in that, particularly when it goes too rapidly or subject to things that trigger irregularities, result in a specific kind of damage that you and I refer to as a tumor. And they even have a specific name for cancers, and not all tumors are cancers, by the way. And they are, if they're in epithelial cells, they're called carcinomas. The most common cancers are all carcinomas. Breast cancer, prostate cancer, which are the most frequent by number, skin cancer, perhaps statistically the most, skin cancer. All of those, remarkably large amounts. Women, about talking maybe 1 out of 7 get breast cancer, maybe men 1 out of 8 get prostate cancer, 1 out of 5 probably for skin cancer. Not all necessarily tragic diagnoses, but potentially they're there. So those are all cancers of lining cells. When the next topic is connective tissues, they are still, relatively speaking, mitotic to replicate, Some of them they can replicate, relatively speaking, completely. Some not. They do form cancers there. They have another name, and it's a little more ominous cancer because it's a cell. It's a lot harder to to replicate and deal with. And typically they're internal sarcoma. So if you ever wondered why we call something a carcinoma and one a sarcoma, it has to do with the cell line or tissue line that is involved. Very interesting. And it's something we all have to either deal with directly or through friends, family members, loved ones, etc. Those are territory. Okay, muscle and nervous we do a whole unit on in the lecture component. I'm not going to get crazy about it. It's just showing you where these tissues are. I don't love that slide anyway. Okay. You're going to visualize the micrographs. You're going to see both schematic representations and actual micrographs, actual microscopic slides here. Some of them with electron microscopy. So what do? You have to, once you get that piece of that sample, it has to be what they call fixed in some kind of preservative agent and then sliced. And we do call them slices. And then stained. The interesting thing, and I'll point that out to you, some things are what we call artifactual. That's a medical term and a scientific term you will hear a fair amount. So, for instance, you'll read an X-ray report or a scan report, and they'll make a comment on something and say, "Note, this may be artifactual." It may have to do with the processing. We saw a lot more of it before we went digital. Okay, now when I started taking X-rays, and I developed them in a tank, dipped them, just the way we used to do the old photography developed. Sometimes it didn't develop properly, and you got imperfections or irregularities. Those were called artifacts. I'll comment on some of those, particularly in the skin unit, about where some things are because of the way it stains, and we may have misinterpreted that are there. So in order to see things, we use dyes. Okay, in the electron microscope, they generate that color with heavy metal coatings because of the nature of what happens when they're subjected to that high energy. You begin to get energy being released that will generate certain colors in the spectrum. I'm not going into that. I don't even go into that in micro. When I do I haven't done a micro lab in some time. Right here. For instance, that's an electron microscopy. These are cilia, if you did not know. So cilia are something that we see in limited areas, by far and away the most in your respiratory system. Cilia get damaged by environmental factors in the respiratory system. Factor number one, smoking. Okay, good news, bad news. Bad news, when you're actively smoking, these get damaged, don't work, disappear. They move mucus around, which traps particles and bad guys, so we can get rid of it. We can expel it in some way, shape, or form. That's the bad. The good news, if you stop, they come back relatively quickly. I'm living proof. I told you. I didn't. I told you the smoking history. So I used to get very long colds. This is what they look like. This is called a scanning. The electron beam comes at an angle. You get kind of a three-dimensional view. This is transmission. It's kind of going right through it, perpendicular. So you get more of the ultrastructure. And when you get to micro, you'll see this. But I figured for the sake of it, we would do this. So we're going to look at the covering or lining epithelia, maybe just a brief comment on glandular epithelia. And again, a lot of those cancers I was talking about, like breast and prostate, those are glandular cancers. So you hear the name adenocarcinoma. Adeno means gland. Like in cold season, we have something called an adenovirus. It's the one that typically gives you a cold with swollen glands, hence the name, even though they're lymph nodes. And they do all these different things. These lining tissues protect, things go through them, they can absorb things, they can give off things. If they have little openings in them, they can filter things that are there. They can be utilized, interestingly, for sensory reception. You get the skin, you'll see that. What makes them very interesting is themselves, they have something called polarity. So what it means to be polarity means they have, it's almost like a top and a bottom. The cells are all connected to some type of a base membrane or a basal structure. So they're connected to each other. And imagine this going this way and backwards that way, a big sheet. And then they're connected to the base. So they're sort of locked in place. Most of the time, those are junctions are a combination of something that you're going to learn about in lecture called tight junctions and desmosomes. Tight junctions, they should put in front of that watertight. The more tight junctions, the less water can seep through. There's so many tight junctions, water can't, or water soluble. That's a big deal in anatomy and physiology. Things that can dissolve in water and things that don't. That's the basis of everything, just about. Because we have a membrane that's made out of fat. Some things can get through it, some cannot. Some need channels or passages with it. That's what movement across membranes, all that diffusion. Facilitated diffusion, osmosis, all that. So you find certain areas where water can't get through. Desmosomes are about strength. They're holding the cells, even the cells go through a lot of physical activity. Think of cardiac muscle cells with your heart beating constantly. Those cells don't pull apart because they're latched together. It's almost like riveted together. Because we can see the ultrastructure under microscopy. So they have specialized contacts, some of those. Underlying every one of those is something called loose areolar connective tissue, under where it's attached. That's how they get nourishment. And that's how they basically release a lot of waste products. Because they don't have their own blood. Most of the time we think of something that can replicate rapidly. It has a great blood supply. This has to get it in directly. It comes from the underlying connective tissue. And that underlying connective tissue, as you will see, and you see it really manifest itself in skin, as something called loose areolar, which means airy. Because it's got a lot of space in it for stuff to move around in. Okay? So, but it has nerve supply. And they are very, as I've told you several times already, regenerative. They replace rapidly. Polarity. So if you look at that illustration I had before, they'll show it to you here, and I'll just skip down. I was going to show it to you, but I'll skip down. Can't do it. They used to have another thing in there. Just a little disappointing. So the surface at the base is called basal. The surface up here at the top is the apex, is called apical. So whether it's one layer or it's multiple layers, you have the basal area, and that's what makes it polarity. It's not the same top and bottom or side to side. So polarity means it's not uniform all the way around. So we can identify things, and we use that a lot. Okay, so the upper or free side is exposed either to the surface, like out of your skin, or the lining in your mouth, or okay, the lining in your gut. You might not think of that, but it's in a cavity. Okay? The lining in a blood vessel. That's inside your body, but certainly it's exposed to an opening that, relatively speaking, is hollow. So we have a lot of those hollow tubules for respiratory, for blood supply, genitourinary. We have a lot of those tubules. Okay? So most times they're smooth. Sometimes, and you'll hear this from me a lot, they have adaptations. Adaptations make surface area frequently. Microvilli, which means tiny fingers, are like the edge of that apical border, has tiny extensions, and all this means more surface area. The more surface area, the more things can go, potentially you have surface area because things pass through the membrane. So here you have the same structures, here, this much space. When we open up, now you have all this extra space that's there. That surface area allows you to absorb or to spit out or excrete things relatively rapidly. You see this a lot in the gastrointestinal area, particularly in the small intestine, because nearly all of your absorption occurs, and the final steps of digestion and almost all of your absorption occurs in the small intestine. You get that in AP2. Okay? Microvilli. So you're going to see a lot of interesting structural adaptations that lend to increased amounts of surface area. I showed one to my earlier class, in the advanced class, which is tracking around the same you are, but we're already on skin. And basically, I showed them the way that this adaptation in the layers of the skin creates a lot of surface area that enhances the blood supply to the avascular epidermis, the lining. So that type of thing. So as we move on, so it's like this. So this is the aforementioned gut area. Those cells are not terribly flat. The name that you're going to see that they get is called columnar, because we have ones that are flat, ones that are kind of square, we call cuboidal. Ones that are flat, we call squamous or squamous. And the ones that are elongated like this, we call columnar. So these are columnar. We call them simple because it's one layer. Simple is another word for single. Okay. Whenever you see the term stratified, it doesn't tell you how many layers. It means there's more than one. And right there, you can't really see it from this particular image, even though it's an actual micrograph. That fuzzy area is like, we call it a brush border. That's filled with those microvilli. I like to use simple illustrations with handy materials. It's just me. I'm going to keep going the wrong way. So here are the contacts I was telling you about, tight junctions, desmosomes. They always have underlying them connective tissue. Typically, the basal lamina or basement membrane, that thing they're attached to, is made of collagen fibers, which are a protein, something they can attach to. We have more collagen in our body than any other protein. A lot of, if you're into nutrition and things like that, people eat and drink a lot of collagen supplements, collagen powders. They'll dissolve them. My wife dissolves it in almond milk, for instance, frequently. Chocolate, of course. My first wife didn't like chocolate. I should have had a clue. Just lay on his heart. I go to Chocolate Anonymous. I'm Lou. I'm a chocoholic. Hi, Lou. Sorry, can't help it. Yeah, it's part of the program. It's a 2-step program. See chocolate and eat chocolate. Not a 12-step program. So, and underneath it, there's something called the reticular lamina with the collagen fibers. The basal lamina is just a sheet of other protein. So it's made of that. It reinforces it. Doesn't tear very easily. Creates a boundary. Boundaries are very important, as you will learn. So what happens? So here, when the idea of a boundary, and this is a really important concept, it's how we distinguish between a benign tumor and a cancerous tumor. So I'll give you the most common cancer that we see today, human papillomavirus, which is a wart virus. The same virus, more or less. There's a lot of different subtypes that create a wart you have on your finger or your toe or your foot or your hand. However, when it's in the genitourinary area, it had the ones that tend to affect there, a large number of them have the tendency to become cancerous, certain types. So how do we distinguish? If we biopsy, take a sample, we take the surrounding tissue, and we want to see whether there's something around it like a border, we call it capsule. If it's encapsulated, that means it's, even though it might be thick and pushing underneath, it's still on top, so to speak. When it starts to go through that boundary, okay, now you got a problem. That means it's invasive. It's eagerly seeking out food and doesn't mind destroying otherwise healthy tissue to do that. The term invasive is a very ominous term. It's what we look for when we define how we are going to treat a cancer. So they penetrate the boundary, invade, and it spreads. And sometimes it can spread in far, once it gets in bloodstream or lymphatics all over the place. So it gives you some idea. So no direct blood supply, regenerates highly, and all you have to do is lose a couple for whatever reason. They're used up, they're abraded, they're irritated, something's got, or it's a mechanical trauma, you fell, you scraped yourself, whatever, you cut yourself, that type of thing. Immediately, there's a fancy term that we see in the human body called contact inhibition. Cancer cells lose that. Contact, it's kind of like you guys see. Like, you'll fill up the chairs. And when they're all filled up, I mean, someone's not going to sit on top of somebody else. Right? So, normal cells form a layer. As soon as that layer is filled, they kind of stop growing. Because they're in contact. You're feeling contact with those cells. Versus cancer cells, which ignore that, they start keeping up, going down. When we grow things in cell culture that forms an even layer, like if you take a beaker or a flask, the growth medium, the cells are all right on top. So the contact is big. When they lose that, big problems. There's a whole bunch of little mechanical sensors that impact it. You're going to get to something in the third chapter in lecture called the growth cycle. It's the one that most people only focus on the part that they can see, which is mitosis. Prophase, metaphase, anaphase, telophase, you're going to go, you're PMAC. So you do that. That's not the important. The important part is interphase. What's basically happening there is all of these steps are being, and there's a variety of checkpoints that as long as they function, the cells don't go crazy, grow. Really. It's up and down stuff. I walked the dog twice this morning. Physically demanding. So simple is single, I told you that. Stratified, 2 or more. Shape of the cells. We base it on, if it's a single layer, the shape's obvious. If it's multiple layers, it's the most apical layer. So you're going to see, most famously, our skin is called stratified squamous. It's based on the shape of the layer, the most apical or outermost layer from the base, not the base layer, which has a variety of shapes. So squamous, flattened, they look like scale. The name actually is an offshoot of shingles, like shingles that overlap, like on your roof. And so you see that for me, I think squamous, squished, squash. Anything with SQ works for me. It's not like squishmallows that you can squish and they come up. There's a lot of money selling squishmallows. It's just a moment. Okay. Cuboidal, box-like, a cube, equal on all sides. The other part of this, as I showed you before, the nucleus reflects the shape. The nucleus is also flat. In cuboidal, it's round. In those columnar, it's more elliptical. So not only does it reflect the outermost layer of cells, it also reflects the shape of the cell, the shape of the nucleus also is part of it. Okay? So in stratified, they vary, so we name it by the apical layer. For instance, stratified squamous, where you can see it's rounder here as it goes toward the surface, effectively changing as it gets away from its blood supply, it begins to change or vary. So you see this on the outer area where it's simple, and notice the shape of the nucleus, rather flat. Actually, the best analogy for simple squamous is like a fried egg. So you have a lot of white and you have the yolk in the middle. So the white is all membrane, and what's great about that, things can get through it. You'll see it shortly. Squamous, cuboidal, columnar. Okay? And so we see a ton of simple because it's got all that membrane. Stuff can go in, stuff can go out. And again, if for some reason the cells are not closely bound to each other, it's almost like this. There's little openings. Like that. None of you would ever know this. There were two very famous PBS TV shows for children. One you certainly know, that's Mr. Rogers here in Pittsburgh. I have a great Fred Rogers story. Remember him? Thank you. I feel better. My granddaughter is going to be 22 in December. I don't think she remembers. My younger granddaughter probably does. The other one's called Romper Room. That's not ringing a bell. No, there was a lady who was like a pre-kindergarten on TV, and she'd like have like she called it a magic mirror, and she'd have names of, she knew kids were watching. They wrote to her, and she had a little mirror that was hollow. I could see Jimmy, and I could see Judy, and all that. It was also Pittsburgh based. The show, hey, it was no Teletubbies. I've gone through children and 2 sets of grandchildren. I know my way around kids' TV shows. Moving on. So, not a lot of cytoplasm, mostly membrane. So, where do we see them? In the kidney, the cells are not close together. They act as a filter. Your blood goes through it. The holes are big enough for water and things dissolved in the water to get through, but proteins and big cells can't. And it creates something magical called filtrate that our bodies resorb 99% of, and what's left is waste product, and you and I call it urine. The lungs, this is the framework of the human lung. The lungs are a very interesting area. You have one membrane in the middle. You have a layer of squamous cells here, and they share that base membrane. So this is in the lung, this is in the blood, and it's all membrane, and the nuclei are relatively small compared to the amount of surface area. And so what happens? Oxygen goes this way, carbon dioxide goes that way, simultaneously. There's one thing about diffusion, if one thing's diffusing, it's not like lung going, you have to wait, come back the other way. More than one molecule, multiple molecules can go in one direction and then the other direction. The human lung is a classic example, and it happens instantaneously, fraction of a second. And so you see it in a lot of different places. Those linings I was talking about, pleura, peritoneum, etc., lining of the blood vessel, lining of the heart, inside. And this is what a lung looks like. Those are chambers that are filled with air. These are the squamous cells, where it's a little more dense. There's connective tissue, maybe some blood vessels like over there. Okay? These are called, the air sacs have a fancy name you'll learn about next term called alveoli. The best example of what an alveolar cluster of alveoli is a puzzle ball, if you like those. It's like a flexible puzzle ball which you can make round. Okay, and it's like all the cells are surrounding it. Does those analogies help? Or else you just, I hope so. And that's the only way I can help you learn this, I think, is to remind you to share these little tricks that help me learn over time. So anytime I can use some type of creative analogy and inject some degree of humor, though none of you seem to appreciate it in this class. You forgot rule number 1. No matter how bad his stupid jokes are, we laugh. Rule number 2, whenever he says, I'm so old, you're supposed to go, no, you can't be that old. You already missed an opportunity. I have a 22-year-old granddaughter. Impossible. Inconceivable. But no, all of you just say, oh, yeah, WTF, he's probably got one, maybe two, right? That's what you're all saying. Oh, help me. So that's, and effectively that's the air. Sometimes when it's, it's a healthy lung. When it's a damaged lung, it gets very thick, and and these chambers interestingly get larger. These cells disappear and they get thick, the ones that are there. So the air gets trapped. Okay, we have a name for that. Trapped air, it isn't only in the lung, it can happen elsewhere. It's called emphysema. So when you're someone with emphysema, it means trapped air. You can have it under your skin. Someone has gas gangrene. They have subcutaneous emphysema. Flesh-eating bacteria, subcutaneous emphysema. Cuboidal. The cell's bigger, the nucleus is bigger, more stuff. It's not just little stuff passing through. Here, we're looking at actively bringing stuff in, actively spitting stuff out. Think more processing. Okay? So glands have a lot of that. The sort of like the glands kind of look like a bottle shape on a rounded bottom, and you'll see it, and most of the cells are simple cuboidal. Typically highlight the rounded nucleus. You'll see it here. There you see that rounded nucleus. This is inside the kidney. The kidney consists, once that filtrate occurs, it goes through a series of tubules. Okay, they start off rounded like this. That's what these are, either proximal or distal, what are called convoluted tubules, because there's a lot of twists and turns. And so it has a lumen, that's the hollow inside, surrounded by these relatively square cells, if you will. And basically what they're doing, that filtrate, they're reclaiming the stuff that you want to keep, and they're getting rid of the stuff you don't want. About 2/3 of the urine is processed right away in the kidney. And it makes its way down, and that's what it's, and if you took a picture of a gland, like a salivary gland, it wouldn't look a whole lot different. When you see columnar, you have a greater degree of processing that goes on. There's more activity as to what it does. So they're taller, and these are when we start to see these projections. I showed you the cilia, now you'll see the microvilli. Cilia, and understand it's not like hair. A microvilli or cilia, and any appendage on a cell is not, it's not like a hair that's sticking out outside of your scalp. They are actually internal appendages. So if you took something that was flexible, like let's say a glove, not 4 gloves. And so if you think of this as the lining of the cell, if I would take something like a cilia or a flagella, it would be an appendage from inside the cell that's sheathed very thinly in membrane. So we call them internal protein appendages. In bacteria, they stick out. But in you and I, they don't. So even though they look like hairs, let's say, they're not. They're actually inside the cell. But the cell, the boundary is so flexible, stuff can protrude. And because of that protrusion, there are certain things they can do more of. So most of where we see these a lot are 2 areas. Basic simple columnar, we see it in the GI tract all the time. Whenever you see columnar, frequently they have cells called goblet cells because they look like a drinking glass, okay, that make mucus. And the other area where you see them, those slightly different arrangement, is in the passageways leading into the lung. They're called, they have a fancy name you're going to get to shortly, called pseudostratified columnar epithelium. Because pseudo means false. It looks like it's multiple layers. It's really only one layer, but not all the cells go to the top. And they all have mucus-producing goblet cells. That kind of sets them apart, where you see columnar. And the mucus is similar in those areas, certainly not identical. And we have a disease that's hereditary that's associated with faults in both of these areas that affect in babies, principally, and very, very young children to start. which identified both respiratory and digestive problems. So for those of you interested in neonatal or in pediatric areas, you're going to see that one of the things when you have a kid who's got chronic respiratory, let's say recurrent respiratory and digestive problems, they look for a particular disease to rule it out. Very fancy name, cystic fibrosis. Because the mucus is, for lack of a better way, overly sticky and stuff gets trapped. So they have problems with digestion and respiration. I know because my middle one was worked up for this at Mercy Hospital near Duquesne. So mucus secreting cells, mucus moves stuff around or traps stuff depending on the area. So, oh no, I didn't mean to do that. It's going to freeze up. So here's the aforementioned gut. Here's that brush border. You can see it a little more clearly. That's a goblet cell, narrow on the bottom, wider at the top. The different staining is the mucus. You can see the columnar epithelia. This is like a pit area. So these little finger-like projections are out there. And then on each of the ones around it, you have microvilli. So you have massive amounts of surface area. They even contain enzymes called brush border enzymes that finalize the product that you're going to absorb. Like it breaks down eventually proteins into amino acids, carbohydrates into simple carbohydrates. So when you hear about somebody who's lactose intolerant, they're lacking an enzyme there to break down lactose, which has got two sugars together, which can't get into the one sugar component of something called glucose and galactose. And it's more common than you think, well, easier to deal with for the most part. So here's what they look like, a nice sheet with all those things. So that would be typical of the gastrointestinal line. We're pseudostratified. It looks like multiple, and the cells typically have cilia because they're moving that mucus towards your mouth to all the stuff that you breathe in that gets trapped. Upper respiratory, some other areas. Genital urinary have ciliated cells because they help to either move the egg along into the oviduct, okay, or also the fallopian tube, same thing, or move sperm effectively, or increase the ability of sperm to move into the vas deferens and eventually be ejaculated. So wherever things have to move along, typically with mucus around them, they help. You want to think of it that way. So they're the cilia. And so, and really the highlight is certainly rest, but you can see you have the cells, this cell doesn't go anywhere near the top. This one narrowly does. This one, the nucleus is higher, but they all have their base at the bottom. And you can see the abundance of goblet cells. That's the interesting thing about it. And so when we're almost done. When we get to stratified, you'll be happy to hear that. Stratified, 2 or more layers of cells. New cells regenerate from below, okay? And what's interesting is it's sort of counterintuitive. When you think of a new layer forming, so here's a layer of cells, you would think the next layer would go right on top of it. Like you got a brick wall, like down here, the blocks here, we put one down, now we're going to put the next one down. It's like, here's the layer that's there. The new layer is going to be underneath it. And then the next layer is going to be underneath that. So we keep pushing it. It's like we're lifting the wall up every time we have a new layer of cells, which would be difficult for a brick wall. But we're pushing it farther away from the base. And in doing so, cells change because they're moving away from their blood supply. When you get to skin, you will see the outer layer is so far from the blood supply, effectively it dies. So they regenerate from below and they move effectively toward the surface. Stratified squamous everywhere. Free surface is flattened, deeper is cuboidal or columnar. High wear and tear, thick layers because there's a lot of pressure. There's a lot of stuff that's going on them, pushing on them, moving over them. So skin is called keratinized because the cells die and become a structural protein called keratin, which we get to in skin. Where in non-keratinized, the difference between your skin here, delicate though it is on your face, to your lips. Keratinized, non-keratinized. Think of any opening, from external to the nose, internal to the nose, or genitourinary openings, anything like that. So and again, high wear and tear areas. So here you can see it. This would be, let's say, in your mouth arbitrarily. The blood supply is from down here, this loose connective tissue. These are the lining cells. So they start here being rather abundant and cuboidal or columnar. As they go farther away, the nuclei flatten, and you can still see these flattened cells. That's why we call them squamous. I grew up on squamous, that's why you pronounce it that way, on the outside. But you can see the nuclei are there. So if you're like me and addicted to crime TV shows, I have an affection for British crime. The British have an affection. So what do they do when they want a DNA sample? You don't have to do a blood test. I do this in my microwaves where I run. I take a sterile Q-tip, stick it in here, and if you took this and put this on a microscope and fixed it, which means heat it slightly so it would stick there, and put some stain on it, you would see epithelial cells. And because the cells are nucleated, they have DNA, and you can get DNA and DNA test with it. That's where you go. The crime shows are having, oh, let me give you something to drink. You have the can. They sneeze or cough, crap out of the garbage. You're not looking. You know, cigarette butt if they find it to see, because you would have it in saliva or mucus or anything. Make sense? So the difference in skin, which I don't think they have pictured here, is those cells are all dead, so you're not getting anything out of it effectively. So and stratified cuboidal, which are 2 layers, or columnar, very rare. Some glands at the base of a gland or the base of the neck of a gland will sometimes see these sweat glands, mammary glands, very limited, urethra, glandular ducts. I mean, there's not a whole lot to say about it. So, I mean, I don't even get into it. And then lastly, and to me, as important as any, transitional. Transitional is only found in what's called the urinary tract. The urinary tract is by definition not the kidney. It's the structures that exit the kidney that contain urine. The ureters, which come out of the kidney, which come together at the bladder and exit the bladder by way of the urethra. Once they're in the urethra, they rapidly transition to stratified squamous, because now you're getting to what I call the outside world. You're getting to the exterior. So, and why they call them transitional, it's an optical illusion. It's not like they magically change. They've got about 7 to 12 layers. When the bladder is filled, where the ureters are filled with urine, it compresses down. It looks like 2 or 3 layers. When the bladder is empty, or it's empty, it transitions back up to its normal appearance. So, basal layer of cells are cuboidal or columnar. They change shape. It's because they can flatten. It's not anything magic. It's about storage. How do you know they're not the same as squamous, though they appear similar to simple squamous? Is you can see that rounded top. Squamous are flat, the nuclei are rounded, like that. So they are really important in that way. So these are all, so this would be an empty, because when you see the rounded top, it's empty. When it's full, you have only 2 or 3 layers down here. And so that's what you see. And then this is the connective, here's the membrane, and here's the underlying connective tissue. So in my case, my bladder cancer, which is where my cancer was discovered in 2015, identified then, was there, and it's classified as what's called TEP1, transitional epithelia type 1, which means it's not just here, but it's kind of like down here, almost at the transition zone. If it was any deeper, it would have been more of a problem to deal with. So it didn't require a great deal of what I would call major treatment. Majors from perspective. Everything's major if they're doing it to you. So anybody say, oh, it's minor surgery. Not if they're doing it to me. It's major. So that, and 99% of the people with bladder cancer have the same thing in common: cigarette smoking. Glands, we classify it based on their secretions. We talk about now is exocrine. They have a duct or a passageway, like a sweat gland to your skin, or a sebaceous gland to the hair and to the follicles that's coming out. The pancreas is a double duty gland. Its exocrine function is a passageway that creates digestive juices to help you in the small intestines to process your food. It also, on its outside, is surrounded by blood vessels that seep up an endocrine secretion, of which there are two, insulin and glucagon, which are vital to your sugar metabolism. And irregularities in that principally manifest themselves as diabetes. So we are concerned now with exocrine, duct glands with a duct, versus endocrine, which are by definition ductless. Okay, so goblet cells are a unicellular exocrine gland. It's one cell, but it's a gland because it makes mucus. Typical salivary gland, multiple. And so it's kind of what they look like. The endocrine glands are surrounded by a nest of blood vessels, and it's like a sponge. It sucks up the secretion. This is a typical gland that you would look at, where it has here's the duct leading out to the surface. If it was a sweat gland, or maybe into what we call the lumen, the hollow area, let's say of your stomach. Okay, and we'll get to that. You have a whole chapter on endocrine in AP2. Exocrine are released onto surfaces like skin or internal linings. Many more than endocrine. They can be uni, which is the goblet cell, or multicellular, and they're classified based on the secretion. Mucus has a sugar protein called mucin, make it sticky. Stain so you can see the secretion's ready to come out. Multicellular are characterized by a couple of things. Mode of secretion is important for us, structure is not. And I'll show you, because they come in what's called tubular or alveolar and tubulo-alveolar, and all these crazy shapes. And in advanced anatomy, which I do in my advanced group, or medical school or professional school anatomy, you do all these. It's all the same. It's just a degree of detail. You're getting the basics. Advanced is getting more detail. Med school, professional school, grad school, a lot more detail. This there. And then the last part for this is the mode of secretion. That's important. And it's all about what it's making. So what it's making is whether or not it's liquid or it's oil or it's something in between. So if it's sweat, that goes by the name merocrine or eccrine. All those different kinds of sweat glands. So you have that same sort of tube shape. You have the cells here. And mostly they're cuboidal, you can imagine circular nuclei. And what they do is they secrete. They just go up little droplets of basically a liquid with some stuff dissolved in it. You and I call it perspiration. Some salt, some other chemicals, some waste products, and out it goes. I mean, you know what it's like outside today. It's hot. Unpleasant. Makes me rethink retiring to Florida. Just a little bit. Okay, that's an eccrine or merocrine secretion. The opposite would be the whole cell being sloughed. They make a new one, and the whole cell, that's called holocrine, which means whole cell. So that's got a lot of stuff. It's got membrane, it's got a whole cell there. So that's very oily, very thick. Sebaceous glands, like we have for our hair. And all of our hair, we have sebaceous glands. They're the ones that unfortunately give us acne when they're filled regularly. And genetically, some people have oily hair, some people have drier hair. You know, you can deal with that, with other topical things. The interesting one to me is the in-between. Accumulate products within, but only the apex, that's the name, apocrine glands. To me, it's not controversial whatsoever. Kind of looks like this. Same deal, and they have an illustration which kind of looks more like this for holocrine. It's like a little fraction, like this little top of the cell comes off. So it's not as much as a whole cell, so it's not as oily, but it's certainly not liquid, it's somewhere in between. Most of us, the way I learned it, so mammary glands for a lactating female, that lactated substance is probably most likely an apocrine secretion. Apocrine, because it has a larger oily component, we also think has to do with the glands that develop anogenitally at puberty. Okay, where they tend to have, they're different in their components and perhaps the odor, or maybe that's affected by genetically or hygiene in certain things. So that's, to me, that's much more the prevailing theory. I don't think it's controversial. So, and again, this is the whole thing with PowerPoints. I don't know who wrote it, but if they think it's controversial. So here's the Americrine or ecrine. They call this holocrine, but to me that looks like fragments. So I'd go the other way. It's just me. And that's just it. Okay. So I need to get you, we'll get the connective tissue doesn't take as long because there's only a handful of them, really like 9 or 10 to look at in there, and they're well grouped. And we should get ourselves into skin. And I want to get that all done. So not next week, but by the 4th week, we can get into bones. So thank you very much. And hopefully both these recordings made their way